Hello all. Today's topic is antigen and antibody. So I'm going to talk on antigen and antibody. So first is antigen. Antigen is a substance that can be that can produce a specific immune response when it introduced to the tissue of an animal that can specifically bind with antibodies or sensitize a cell is known as an antigen, and the ability of antigen to produce immune response and react with the product is known as antigenicity. So ability of material to induce an immune response is referred as immunogenicity, and such material is known as immunogens. So epitopes and paratopes are the two terms that comes in a picture. So epitopes, the part of antigen with which antibody react, is known as epitope or antigenic determinant, and the portion of the antibody molecule that bind to the epitope is called as paratope. So epitope determinants is bound to the specific antibody by non-covalent bond, and their binding is more or less three-dimensional and may be similar to lock and key model. So there are some essential factors of antigenicity. We can call this characteristic of antigen. So its size, the molecular size of antigen is directly related to the antibody. So antigenicity, so very large molecule such as hemocene or tetanus, they are excellent antigen. The larger the size, they are excellent in that way. So chemical nature, most of the naturally occurring antigen are proteins and polysaccharide. So these antigen are found to be comparatively more antigenic than the lipid and the nucleic acid one. Then comes in solubility, the non-antigenicity of synthetic polymer also attribute to their insolubility in body fluids. So next is foreignness. Foreignness is very important. So antigen is a macromolecule that must come from a foreign sources. There are some exceptions. So these antigen are related species are less antigenic than that of unrelated species. This is all the characteristic of antigen. The next come antibody. Antibody is y-shaped. It has a y-shape. Antibody is also called as immunoglobulin. Immunoglobulin are immunologically active serum protein that is formed in response to the antigen, and that react specifically to that antigen only. So the blood protein produced in response to and countering a specific antigen. So antibodies combine chemically with substances that body recognize an alien, such as antigen. That antigen can be bacteria, viruses, foreign substances like pollen as well. So antibodies are y-shaped protein, and they play pivotal role in immunity against some viral and bacterial infection. So the structure of antibody, it is y-shaped. It has two chains, heavy chain and light chain. This heavy chain and light chain have hinge region. Means the heavy chain have hinge region that is very flexible. So they move according to the antigen, and they both are bind together by disulfide bond, and this antibody have antigen binding site in the top of them, and they also have glycosylation site. So there are types of antibody. So antibody is also called as immunoglobulin. So immunoglobulin are of 5 types: immunoglobulin A, D, E, G and M. So about immunoglobulin G. We will start with most prominent type of. So immunoglobulin G, it is predominant, and normally it contains 85% of serum. So this is the only immunoglobulin that can be transferred from mothers to the fetus through placenta, and they play important role in hemolytic diseases of newborn. And they are smallest, like 1,50,000 mw. So it is capable of binding to the complements, and it is produced predominantly in secondary immune response. Then comes IgM. IgM is present in 10% in the serum, and it is pentameric structure. It has pentameric structure. It has largest molecular weight, that is 9 lakhs, and because of its larger size they cannot pass through the placenta, and it is generated in the primary immune response. Then comes an IgA. IgA molecular weight is 1,60,000. It is present in 10 to 15%, and it is the only antigen that is present in secretion like saliva, or tears, or cholesterol and breast milk. It is not going to transfer in the human placenta, and it does not fix to the complement as well. So this is all about IgA. Then comes an IgD, that is immunoglobulin D. It activates B lymphocyte. It binds to the B lymphocyte. It is present in 1% only, and it is very susceptible to proteolysis. So IgD present on the surface of B cell, and they play important role in induction of antibody production as well, and prevention of respiratory tract infection as well. So the last one is IgE, that is immunoglobulin E. Immunoglobulin E is a monomer that is present in very less quantity, like 0.004% in blood plasma, and it activates only in allergic condition or hypersensitivity. It causes the release of histamine and heparin from the cell, and it binds strongly to the receptor on the mast cell or basophils. So this is all about immunoglobulin present in our body. So immunoglobulin. Then I am going to talk on properties of this immunoglobulin. Immunoglobulins are glycoprotein. They have Y shape or T cell and T shape. Molecular weight can be ranges from 1,50,000 to 9,50,000, and immunoglobulin molecule is made up of polypeptide chain. There are 2 light chains and remaining are 2 heavy chains, and they cross placenta, like that is IgE. They have reagenic activity, that is IgE. They involve in complex fixation, that is IgG and IgM, and they fix macrophages, that is IgG, and they also bind with the mast cell or fix basophil, that is IgE, and they also agglutinated antigen. So this is all about antigen and antibody. Thank you.